Hi everyone, we are going to continue learn about the lipid. Membrane formation as a consequence of amorphic nature of the molecules, and their polar head groups favor contact with water, whereas their hydrocarbon tails interact with one another in preference to water. So how can molecules with these preferences arrange themselves in aqueous solutions? One way is to form a globular structure called a meso. So the polar head groups from the outside surface of the meso, which is surrounded by water, and the hydrocarbon tails are sequestered inside, interacting with one another. Alternatively, the strongly opposite preferences of the hydrophilic and hydrophobic moieties of membrane lipids can be satisfied by forming a lipid biolayer composed of two lipid sheets. A lipid biolayer is also called a biomolecular sheet. The propensity of phospholipids to form membranes has been used to create an important experimental and clinical tool. Lipid vesicles or liposomes are aqueous compartments enclosed by a lipid biolayer. These structures can be used to study membrane permeability or to deliver chemicals to cells. Ions or molecules can be trapped in an aqueous compartment of lipid vesicles by forming the vesicles in the presence of these substances. Liposomes can be formed with specific membrane proteins embedded in them by solubilizing the proteins in the presence of detergents and then adding them to the phospholipids from which liposomes will be formed. Protein liposome complexes provide valuable experimental tools for examining a range of membrane protein functions. Permeability studies of lipid vesicles and electrical conductance measurements of polymer biolayers have shown that lipid biolayer membranes have a very low permeability for ions and most polar molecules. Water is a conspicuous exception to this generalization. It traverses such membranes rapidly, easily, because of its lower molecule weight and high concentration and the lack of a complete charge. The range of measured permeability coefficients is very wide. We now turn to membrane proteins, which are responsible for most of the dynamic processes carried out by membranes. Membrane lipids form a permeability barrier and thereby establish compartments, whereas specific proteins mediate nearly all other membrane functions. In particular, proteins transport chemicals and information across a membrane. Membrane lipids create the appropriate environment for the action of such proteins. The protein components of a membrane can be readily visualized by SDS page electrophoresis. The gel electrophoresis patterns are three membranes: the plasma membranes of erythrocytes, the photoreceptors membrane of retinal rod cells, and the cytoplasmic reticular membrane of muscle. It is evident that each of these three membranes contains many proteins, but has a distinct protein composition. In general, membranes performing different functions contain different representatives of proteins. The ease with which a protein can be dissociated from a membrane indicates or intermittently it is associated with the membrane. Some membrane proteins can be solubilized by relatively mild means, such as extraction by a solution of a high ionic strength. Other membrane proteins are bound much more tenaciously, so they can be solubilized only by using a detergent, or an organic solvent. Membrane proteins can be classified as being either peripheral or integral on the basis of this difference in dissociability. Integral membrane proteins interact extensively with the hydrocarbon chains of membrane lipids, and they can be released only by agents that compete for these nonpolar interactions. Membrane proteins are more difficult to purify and crystallize than water-soluble proteins. Membrane proteins differ from soluble proteins in the distribution of hydrophobic and hydrophilic groups. We will consider the structures of these membrane proteins in some detail. The first we considered is the archaeal protein bacteriorhodopsin. The protein uses light energy to transport protons from inside to outside the cell, generating a proton gradient used to form ATP. Bacteriorhodopsin is built almost entirely of a helix. 7 closely packed alpha helix, nearly perpendicular to the plane of the cell membrane. Examination of the primary structure of bacteriorhodopsin reveals that most of the amino acids in this membrane-spanning alpha helix are nonpolar and only a very few are charged. This distribution of nonpolar amino acids is sensible because these residues are either in contact with the hydrocarbon core of the membrane or with one another. Porin, a protein from the outer membrane of bacteria such as E. coli, represents a class of membrane proteins with a completely different type of structure. Structures of this type are built from beta strands and contain essentially no alpha helix. The arrangement of beta strands is quite simple. Each strand is hydrogen bonded to its neighbor in anti-parallel arrangement, forming a single beta sheet. This arrangement of nonpolar and polar surfaces is accomplished by the alteration of hydrophobic and hydrophilic amino acids along each beta strand. The membrane proteins considered thus far associated with the membrane through surfaces generated by hydrophobic amino acid side chains. However, even otherwise soluble proteins can associate with membranes if hydrophobic groups are attached to the proteins. 3 such groups are shown in the slides. These modifications are attached by enzyme systems that recognize specific signal sequences near the site of attachment. Many membrane proteins employ a helix to span the hydrophobic part of a membrane. As noted earlier, so most of the residues in this alpha helix are nonpolar, and almost none of them are charged. Can we use this information to identify likely membrane-spanning regions from sequence data alone? One approach to identifying transmembrane helix is to ask whether a postulated helix segment is likely to be more stable in hydrocarbon environment or in water. Specifically, we want to examine the free energy change when alpha helix segment is transferred from the interior of a membrane to water. Free energy changes for the transfer of individual amino acid residues from hydrophobic to an aqueous environment is given in the table. The hydrocarbon core of a membrane is typically 30 angstrom wide, a length that can be traversed by an alpha helix consisting of 20 residues. We can take the amino acid sequence of a protein and estimate the free energy change that takes place when a hypothetical alpha helix formed of residues 1 through 20 is transferred from the membrane interior to water. For example, glycophorin, a protein found in the membranes of red blood cells, is predicted by this criterion to have one membrane-spanning helix in agreement with experimental findings. Okay, here is some knowledge about lipids for today, and thank you for your listening.